The cell cycle is regulated with checkpoints, which are controlled by cyclins and cyclin-dependent kinases, but mutations can cause the cell to die through apoptosis or possibly could cause cancer. So as we look here, we have our cell cycle. We have our interphase, we have our mitosis, and we have our cytokinesis. And we have three major checkpoints that we consider here. During this G1 phase, we have the G1 checkpoint. And in the S phase is when their DNA replication is going to occur. So before this period starts and the DNA gets replicated, it's first checked to see if there's DNA damage and if there is enough nutrients in the cell is big enough. So again, if there's any problems here, the cell will be programmed to be destroyed and if it can't be fixed. Now, after that DNA is replicated, there's another checkpoint before the nucleus gets divided. And again, this checkpoint here is going to check for DNA damage and it will try to fix it. And again, if it can't be fixed, it will be destroyed. In this checkpoint, the metaphase, at the metaphase checkpoint, you're going to check to make sure all the spindle fibers are attached correctly and the chromosomes are aligned. And again, it's not going to continue if we're going to get some non-disjunction. So we have three main checkpoints that we're considering here. With these checkpoints, it's important to understand that it's genes that are controlling the checkpoints. And the two main categories of genes that we have are tumor suppressor genes, which again, go through this interphase and mitosis. As it's going through G1, you're going to hit a stop sign here. The tumor suppressor genes are going to stop the cell cycle, fix anything that needs to get fixed, or destroy it if it's unfixable. After it gets through that checkpoint, again, this checkpoint is like, is the cell big enough? If it can say yes, and the DNA is without damage, then proto-oncogenes are going to be the gas pedals to push it through to the next phase. And the proto-oncogenes might make more mitogen receptors, more cyclins, and we'll get into what those are in just a sec. So again, as it keeps going, tumor suppressor genes stop, proto-oncogenes go. Tumor suppressor genes stop, proto-oncogenes go. So when we're looking at these different things, again, the cell is going to get a signal, which might be a mitogen or a growth factor, that it's going to stimulate the proto-oncogenes to make proteins that are going to push the cell towards division. Now, at that same point, you also have these tumor suppressor genes, which are like stop signs or brakes, which are going to pause the cell cycle and correct things that are wrong or destroy damaged cells. So these tumor suppressor genes, P53 is a really commonly discussed tumor suppressor gene. The P53 protein ends up being a transcription factor. Now, if P53 protein is hanging around, it's not just active all the time. It might be in an active form, but if no DNA damage is recognized, that protein will break down over time. But when DNA damage is observed, the P53 protein becomes phosphorylated. Again, we attach a phosphate group, which now makes it active, which then, as a transcription factor, it can attach the promoter region to help turn on genes that can repair. Now, if DNA damage continues to be observed and observed and observed, then and it isn't getting repaired, then that P53 phosphorylated protein will also turn on genes that will cause apoptosis. And apoptosis is programmed cell death. This diagram demonstrates what's going on. The cells round up, the nucleus collapses, and ultimately that chromatin in the DNA starts splintering apart. And we have these fragments, and then you get these blebs, and those blebs are going to kind of segregate or split off parts of these fragments of the nucleus, which is going to cause this entire cell to die. Now, on the other side, when we have brakes and all of that, we also have the gas pedal. And the gas pedal is caused from proto-oncogenes. So we have interactions between cyclins and cyclin-dependent kinases. So typically the cyclin-dependent kinases are going to, again, phosphorylate things, but they'll kind of be around. But these cyclins go in waves. And when the cyclin, you get enough cyclin present, it's going to turn on genes that are going to move it into the next phase. So again, you have the cyclin-dependent kinase. Once you have the cyclin in a high enough concentration attached to the cyclin-dependent kinase, it becomes phosphorylated. And this kinase can phosphorylate other proteins, which again turns them into active forms, which can then again become a transcription factor and turn on other proteins, which will push them through the phases. So again, another way of looking at this, proto-oncogenes, you have problems that can arise. So if a proto-oncogene becomes mutated, you still have this information coming in, and it's going to turn on this oncogene, and that a proto-oncogene, when mutated, becomes an oncogene, a cancer gene, and that's going to cause more protein to be made, more receptors. So every single growth factor is going to keep on sending the signal pathways to go, 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 go. Or it might make, this oncogene might make more cyclins, so the concentration stays high. Or it may make a slightly deformed form of the cyclin, which can still bind to the cyclin-dependent kinase, but it isn't broken down on its own, and they don't degrade. So they can ultimately, an oncogene means like the gas pedals are stuck, and it's going to keep pushing it through the phases of the cell cycle. Tumor suppressor genes, when they are mutated, that gives us a mutated tumor suppressor gene. And the protein that's supposed to be pausing or fixing or causing apoptosis can no longer do that job because that is mutated. And that protein, ultimately, it's like saying that there's no brakes and there's no construction workers. So when we start looking at this cycle, if you have tumor suppressor genes being mutated, it means you can't stop, you can't fix, and you can't destroy those bad cells. So ultimately, you're getting rid of all of the stop signs. And then when you have oncogenes, you just get all gas pedals and more mitogen receptors, more cyclins, just pushing through all the checkpoints without any regulation. And that's what cancer is. It's unregulated cell division. So when we start thinking about cancer, again, you get a bad cell made. And again, not every mutation causes cancer. It has to be mutations that exist within the tumor suppressor genes or the proto-oncogenes that are regulating the cell cycle. So you start getting this primary tumor. Now, more mutations take place. Typically, cancer isn't just, one mutation, now it's going to become cancerous. When you start not regulating and not checking or fixing the DNA, more and more mutations can occur over time, and then it can spread. It can grow out of the tissue that it started from, but then sometimes it can jump into vessels like your blood vessels or your lymph vessels and can spread to new areas of the body, typically far away, possibly from the primary tumor where it initially started. So again, cancer cells are not differentiated. They don't do the job that they're supposed to be doing. Normal cells have jobs to do, and they're designed to carry out that job. Cancer cells aren't carrying out any jobs. They're abnormal nucleus. They don't break down. They won't kill themselves when they're messed up. If they're pushing against something, normal cells won't divide if they're butted up against another cell. These guys, they'll continue to grow and crowd out healthy tissue. Again, normal cells are organized, cancer cells are disorganized, and normal cells will remain where they're supposed to be, but cancer cells can grow and spread. So checkpoints are a lot of times the regulation. It has to do with cyclin proteins, and if the checkpoints get messed up, cells can die, which is actually a good thing. Or if they don't destroy themselves, you could get cancer.